start the recording. Now let me share the screen. Okay, are there any questions? So if we look at what's on the schedule for today, it's just lecture, catch up, and review. I think in the second part of the lesson I'll begin chapter 7. But for the moment, I'm just going to answer questions and then do some review for your quiz. Any questions about that? Okay. How far did I get on chapter 13? Does anyone remember what slide I got in? I remember I talked about that one, and I talked about that one. Have I talked about that slide?
Two more, I think.
Two more, okay. So I talked about that one. So I talked about this one, and I do remember I was talking about nucleic acid. So I think I interrupted on that slide. If you don't want to listen to the review, you can come back in the second part of the lesson. I'll start up on chapter 7, but for the first part of the lesson, I'm just going to cover review and answer any students' questions. Let me turn the volume up a little bit. How am I coming in now?
You sound fine.
Okay, good.
Perfect. Perfect.
All right, so we mentioned that the genome in a virus can be either DNA or RNA, and that DNA can be either double-stranded, and the RNA as well, or single-stranded. Now, when we're talking about single-stranded RNA, remember you have to distinguish between positive single-stranded RNA and negative single-strand RNA. The positive single-strand RNA genome can be read by the ribosome, and the negative single-strand RNA genome cannot be read by the ribosome. Any questions about that? Viruses, besides their genome, will be surrounded by a protein coat. We call that the capsid. The different subunits of the capsid are called capsomeres. If there's no envelope, it's called a naked virus. And if it has an envelope, then we call it an enveloped or enveloped virus. Only some viruses have an envelope, and these are the viruses that have the budding life cycle. Any questions about that? Envelope viruses tend to have spikes on them. Let me blow this up a little bit. And if there are spikes, those are the outermost regions of the virus. And so the outermost regions of the virus are what the host immune system recognizes as antigens. And so for a spiked virus, the spikes are antigens. I haven't really defined what antigens are, but it's what the host immune system recognizes as being foreign. And they're the most external parts to a virus or another entity which is invading the human body. So the external molecules. When we're talking about virus structure, it's important to realize that we're talking about general viral shape, its morphology, and viruses take on three shapes. Helical viruses, where the capsid is actually in a helix. Polyhedrons, where the virus is many-sided, like a many-sided dice. And then complex virus, where the virus can take on essentially any shape. Okay, any questions about that? You should know the virus T4. It has the shape of a lunar lander. And why you should know this virus is because it's the virus that's been studied the best, the most. I mean, it's probably just because that virus grows in E. coli. This virus is unusual in that it has different capsomeres for the different region of the virus. There's the head region, which is the main region of the virus containing the genome, and then a sheath and a tail base plate, and then a tail fibers. The bottom of the tail fibers is where the virus actually attaches to its host cell, and the sheath and the base plate, as well as the rest of the tail part, is a structure for the virus designed to get through the cell wall of a bacteria. Well, yeah, the cell wall of a bacteria, because a bacteriophage, meaning a bacteria virus, has to have something to help it get through the cell wall, and the tail is what accomplishes that. Many, not all, but many animal viruses don't have a tail because it's much easier to infect an animal cell, much easier to get into the cell. You don't have to go through the cell wall. With viral infection, this can lead the host to produce antibodies, and this, we're really talking about an animal here. Bacteria don't produce antibodies to an infection, but when a virus infects an animal, it can stimulate the host to produce antibodies, and the antibodies are the host's immune response to being invaded by a foreign entity such as a virus, and the antibodies tend to recognize the viral surface proteins or glycoproteins, and that we call antigens. And I'm not talking about T cells here, but in the future lesson I'll talk about T cells. And so it can actually be antibodies and T cells that recognize the antigens to the virus. Some viruses have ways to protect against that, and that is their viral proteins change or mutate frequently, and therefore different strains of the virus can infect the same individual because the strains are different, and the host immune system will only react to the strain of the virus that infects you. An example is the human flu, and I'm afraid that COVID-19 is probably one of these viruses. We'll know more later about that. Obviously, COVID-19 hasn't been studied long enough, but it probably will change or mutate frequently. And so the immune system may not respond well to a vaccine on COVID-19. And this is something that will be a real problem once they develop the vaccine. And that is, we may need to have several vaccinations per year per COVID-19. By several, I'm talking about, well, more than one, and at least two, and maybe as many as four. And the question is, will they need to be different vaccines, or will they be the same vaccine? And to that, we don't know the question yet. But it turns out that coronaviruses, when we've made vaccines to them, and they've only made vaccines to the coronaviruses that cause human colds, the vaccines don't work well, and the coronavirus tends to change. And we will see about COVID-19. Okay, so that's still a story that's being studied, but it looks like the vaccine won't be good for long term, meaning where you take one and it'll last your lifetime. When we talk about viruses, we have taxonomy, how viruses are grouped, and all we talked about is that viruses are grouped into families, and then genera under that, species under that, and then viral strains. There are some viruses that we know enough about them that they're actually grouped into orders, but I didn't talk about that because not all viruses have orders. And a group beyond that, we just don't know how to group them. And the reason why is because viruses have a very small genome, and their genome evolves quickly to the host cell that they're infecting. And so we just don't know the evolutionary history of viruses. So they're not grouped above, most of them are not grouped above families. Although, like I said, there are a few that have orders. There is one grouping above families, and it's really the kingdom that the virus infects. And you can use that for the virus as well. And that is, we do group viruses into like animal viruses, so they infect the animal kingdom, and then we have viruses grouped by bacteriophages. They infect bacteria. So we do group them that way, the host cell that they infect. Any question about that? The subspecies are designated by a number, and subspecies are very important for viruses. For example, I'm wearing my wrong glasses here. Viruses in subspecies, one virus that causes polio is the virus that causes the polio disease. And then another polio virus, the subspecies, is the one we use for the live vaccine to prevent polio. So it's a different subspecies, and we use this as a vaccine to prevent polio. So that tells you how different the viral subspecies can be. One causes disease, and the other one we're using as a vaccine to prevent polio. Any questions about any of that? We give viruses family names, and the family names always end in viridae. And then we break them down into their genus. For herpes viridae, simplex virus would be the genus. That's the common name. And then the subspecies type 1, so herpes simplex type 1. And that's how we name viruses. There is no Latin name, it's just the common English name for the virus. We can grow viruses in the lab, and these plaques are showing you viruses that are growing. They're bacteria viruses on a lawn of bacteria growing on this plate. And wherever there's a plaque, there was one virus that went on to lyse other bacteria on the plate. Viruses, of course, hijacked a host cell and use it to replicate. That's pretty much how all viruses replicate, although it is different between a lytic virus and a non-lytic virus. A lytic virus will lyse the cell to escape, and a non-lytic virus will not lyse the cell. So in the non-lytic virus, the cell can survive, but in all cases, the virus hijacks the cell and then uses the host enzymes and components to make new viruses. For bacteriophages, there's two life cycles, the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, the host cell is killed, releasing viruses. In the lysogenic life cycle, the host cell remains alive. And when the virus is replicating, it needs to replicate its host genome and then translate that genome into messenger RNA, viral messenger RNA, and translate that into viral proteins, and then make the new virion by putting the capsid proteins together with the viral genome. The lytic life cycle goes through 5 stages: attachment, penetration, biosynthesis, maturation of the new viruses, and then release. So in the lytic life cycle, the virus attaches to the host cell. It then contracts, and that will put the viral genome injected into the host cell. And I should mention that in the attachment phase, the virus opens up a little portion of the cell wall so that the virus can actually inject its genome into the cell. Getting through the cell membrane isn't difficult because the structure of the virus just essentially punctures the cell membrane, and then the host genome is injected into the cell. The genome then be transcribed. The first thing it does is, and translated, it'll create an enzyme which destroys the host chromosome, and then the virus replicates its genome, translates its viral genome into proteins, and then there's assembly period. The new virions will be assembled, and the last thing the host cell or the viral genome does to the host cell, it lyses the host cell, and that releases the new genome. So the burst time, the time from infection of the host cell to the time when the host cell burst, the burst size, the number of new virions released. Any questions about any of that? That's species dependent, meaning the viral species, on what the time is and what the burst size is, burst time and burst size, and it also depends on the temperature. Lysogenic virus can follow the lytic life cycle if, when a lysogenic virus infects a host cell and it decides to undergo the lytic life cycle, in which case it just goes through the lytic life cycle where the virus replicates and bursts the cell, and then the new virions go out and attach to new bacteria. So that's the lytic life cycle. But the point is that when a lysogenic virus first infects a cell, it has to determine, will it go into the lytic life cycle or will it go into the lysogenic life cycle? And if it goes into the lysogenic life cycle, there's a repressor protein which is translated, and that's the lysogenic life cycle, and the repressor protein shuts down the genes of the lytic life cycle. Any question about any of that? If it enters the lysogenic life cycle, the genome of the bacteriophage incorporates into the genome of the host cell, and that's called the prophage. And the only time the prophage replicates is when the host cell replicates its DNA, and then each daughter cell for this replicated cell will carry the lysogenic virus, meaning the genome of the virus. And that can go on for many cell divisions. Eventually, though, the prophage can exit the host chromosome, and then enter the lytic life cycle. What changes this cell from leaving the lysogenic life cycle to entering the lytic life cycle is not well understood. And they've been studying that now, I would say, for over 50 years. It's probably over 70 years now. And they still don't have a good feeling for why the prophage decides to leave the lysogenic life cycle. Okay. One thing that can happen is if the genome of the host cell becomes damaged, the virus can leave the lysogenic life cycle to enter the lytic life cycle. And they think that the reason why the virus is doing that is the virus knows that the genome has been damaged and that the cell may die, and so the virus enters the lytic life cycle. But other than that, they don't know why the lysogenic virus leaves that life cycle and enters the lytic life cycle. Okay, any questions about that? Excuse me here. Some consequences for the cell having a lysogenic virus is, for one thing, the host cell cannot get infected with other viruses of that species of virus. So that cell is immune to infection by that species of virus. And then another consequence of this cell is that it has a viral genome in it, and that viral genome can be expressed, and that can bring new genes, new phenotype to this bacteria, because it now has more DNA, and it's the DNA of the virus. The only example I mentioned about that is that Corynebacterium diphtherium does not have the diphtheria toxin unless it has a lysogenic virus, and that the lysogenic virus encodes the diphtheria toxin for Corynebacterium diphtherium. So the only way you can have that toxin is if you're infected by Corynebacterium diphtherium that has a lysogenic virus that infected it. Any questions about that? So let's talk a little bit about animal viruses. And I mentioned that they go through pretty much the same pattern of viral... Oh, excuse me, I just sneezed there. The same viral replication, but there are some differences. For example, they attach to the cell membrane because animal cells don't have a cell wall. The penetration is different, and it doesn't happen by the virus putting a hole in the cell wall and then contracting and injecting the genome into the cell. The virus gets into the cell either by endocytosis, or if it has a lipid membrane, it can have its lipid envelope. The lipid envelope can actually fuse to the cell membrane of the animal cell. And then the two, when they're fused, that can cause the virus to get into the cell. Any question about any of that? Biosynthesis is pretty much the same. Maturation also pretty much the same. Release is a little different, and that is it's actually the same if we're talking about a lytic virus. And animal cells do have viruses that follow the lytic life cycle. But there's also the budding life cycle. How a virus replicates is different if it follows the budding life cycle. And I'll talk about that in a little more detail, how the virus if it follows the budding life cycle, that's how the virus obtains its envelope. Another difference is that bacteria viruses always have to replicate in the cell cytoplasm because there is no other place for the virus to replicate. But an animal virus can replicate either in the cytoplasm, and the RNA viruses tend to do that, but the DNA viruses tend to replicate in the nucleus. And why they replicate in the nucleus? Because that is where animal cells have all the enzymes and the components that a DNA virus needs to replicate its DNA genome. Any questions about that? So those are differences between animal virus and bacteria viruses. I've already talked about this, so we're going to move on, move on, move on. Let's come back to this. We did mention that animal viruses, some of them follow the budding life cycle. And when a budding virus replicates, it replicates at low numbers, and the virus will replicate its genome and translate its capsid proteins, and they get together to make the new virion. And then that virion will bud out of a membrane, and it tends to have viral proteins in that region of the membrane where the virus buds out of. And it just pushes on that membrane. This membrane is shown as the cell membrane, but it can be the nuclear membrane or the membrane of the endoplasmic reticulum. It just depends on the species of virus for what membrane it buds out of. And it pushes on that membrane until it rips off a piece of that membrane, and then that membrane becomes the envelope of this budding virus. And there's an actual electron microscopic picture of that happening. And you see these blue regions in that membrane? Those are actually the viral proteins in the envelope, or the glycoproteins in the envelope. And if you remember the spikes on a virus, let's go way back here. Is that a spike? No, well, they're there, but it's not very visible. Let's go back to this one. Those spikes are proteins or glycoproteins in the virus. And those are encoded for by the genome of the virus. And so they're in the membrane that's ripped off when the budding virus forms. But the glycoproteins, or the spikes, the proteins, are actually encoded for by the virus. Any question about any of that? And that's how a budding virus obtains its envelope. The point is the budding virus buds out of the cell at low numbers, and so the budding virus does not usually kill its cell. If the cell dies, it's because the host immune system recognizes that this cell is infected, and so the host immune system kills the cell. Okay, any question about that? For the DNA viruses, you don't need to know the DNA viruses, but here they are here. That's way too big. Sorry. Tried to blow it up and it blew up really big. There we go. As I mentioned, the DNA viruses replicate in the nucleus of an animal cell. And there are no exceptions to that one that I know of. Viruses, especially the DNA viruses, they can, if they enter the lytic life cycle, express the viral genes, reproduce new viruses, and then lyse the cell. So the genome is replicated, the capsid proteins are made, the new virions are replicated, and then the virus lyses the cell and that releases the viruses. But animal viruses may integrate into the host cell's DNA, and if they do that, the viral DNA permanently becomes a member of that host cell DNA. And we don't call that a prophage, it's called a provirus. That's going through the lytic life cycle, where a DNA animal virus infects the host cell, it's brought into the cell, it gets its genome into the nucleus, and then the genome is transcribed into messenger RNA, which goes into the cytoplasm to make the capsid proteins. The genome, in the meantime, is replicated in the nucleus, and then the genome gets together with the capsid proteins, making a new virion, and then the host cell is lysed, releasing the lytic virus. All right, any questions about the DNA viruses in general? I only talked about it with the lytic life cycle, but we briefly mentioned the budding life cycle. These are the RNA viruses, and they replicate in the cytoplasm of the cell for animal cells, with one exception. I'm not seeing it now. Retroviruses. Let me move this down. It must be lower. Does anyone see retroviruses on there? Oh, there it is, right there. So that's one exception. The retroviruses replicate in the nucleus of the cell. All the other RNA viruses replicate in the cytoplasm of the cell. We'll also talk about the coronaviruses briefly, looking at different RNA viruses. For RNA viruses, if it's double-stranded, you just use one template of the RNA and use it to make the other template. So that's similar to DNA. If it's the single-stranded RNA virus, if you're the positive single-strand RNA, to replicate your genome, you make the corollary strand, the negative strand RNA, and then you use that strand to replicate more of the positive strand RNA, because that's your genome. On the other hand, if you're a negative strand RNA, that's your genome, you make the positive strand, and then you use the positive strand to make more of your negative strand genome. The negative strand cannot be read by ribosomes, but the positive strand can be read by ribosomes. And so, regardless of what the genome is, only the positive strand is put into the cytoplasm to be read by ribosomes to make the viral proteins. Any question about any of that? So here we're looking at a slide of that. How a DNA virus is transcribed to make messenger RNA and then translated into protein. Actually, a double-stranded RNA pretty much follows that same pattern. One copy will be made as a viral messenger RNA and then sent to the cytoplasm to be made into protein, and the single-stranded RNA makes the other copy to make more of your genome, and then only the positive strand RNA is used as messenger RNA to be translated into protein. And then a picture of RNA virus infecting a cell, being brought into the cell. It will be transcribed and then translated into viral protein in the cytoplasm. And if that's positive strand, single strand RNA virus, it doesn't have to be transcribed. That positive strand RNA molecule can be directly read by the ribosomes to be translated into viral proteins. But regardless, that viral genome, you make the other strand to replicate your strand of the genome, and then only the positive strand RNA will be read to be translated into protein. The viral proteins get together with the viral genome to make the viral new virions, and I'm not sure if that's the budding life cycle or the lytic life cycle, but the virus comes out of the cell either by lysis of the cell if it's following the lytic life cycle, or by budding if it's following the budding life cycle. Now, retroviruses are different. This is an RNA virus, but it has the enzyme reverse transcriptase. That enzyme reverse transcriptase takes the genome of the virus, which is positive strand, single-stranded RNA, and makes DNA, eventually making double-stranded DNA from that single-stranded positive strand RNA genome. That DNA then moves into the nuclease, and don't I have a slide showing that? Oh, there it is. That DNA moves into the nuclease, and it then integrates into the host chromosome and becomes permanently a member of that cell. And then for the virus to replicate, what it does is the DNA will be transcribed as viral messenger RNA, which will go into the cytoplasm, and that will make the capsid proteins of the virus. The DNA will be transcribed as positive strand, single strand RNA virus, which will be the genome of the virus, and that viral genome to get together with the capsid proteins to make the new virion. And then the new virion, or naked virus, whatever you want to call it, will push up on a membrane of the cell that has glycoprotein spikes in it, and we'll push on that and we'll rip off a piece of the membrane, and that can be the nuclear membrane or the cell membrane or the endoplasmic reticulum, and rip off a piece of the membrane and that will become the envelope for the retrovirus. Any questions about retroviruses? That retrovirus will become a permanent member of the DNA to this cell. Most of the time, this is not a germline cell, so it's unimportant to the species, but if it infects the germline, that retrovirus DNA will become the DNA to that host species. Humans have it's either 5 or 6 retroviruses in their genome, but the retroviruses fortunately are mutated, and so they cannot replicate the viruses. And that retroviral DNA has now become part of the human genome. Any question about that? All right, that's it for retroviruses. They're unique among the RNA viruses. Coronaviruses are not unique among the RNA viruses, but I talked about them because of COVID-19, the pandemic hitting the world right now, and realized that coronaviruses are more than just COVID-19. They're also SARS and MERS, which are actually more lethal than COVID-19. SARS hit in 2003 and appears to be extinct. Let's hope it is. At least it hasn't hit the human population. And, I don't know if they know where that came from. obviously from an animal. The question is SARS still around, and can it reinfect the human population in the future? And I don't know the answer to that. Whoops, let's undo that. MERS is a virus which is very lethal. About 36% of the humans who get it die. Fortunately, MERS doesn't spread person to person, and it generally comes only from people who are around camels, and you have to be around the camel for a lengthy time before you become infected with MERS. And then the other coronaviruses which hit the humans all the time, especially young children, is coronaviruses tend to cause human colds. Okay? And obviously these hit humans all the time, and they're not a big burden to the human species. But COVID-19 is, and that's why we're talking about coronaviruses. It appears to be 3 coronaviruses that replicated, 2 from the bat and 1 from the pangolin. A pangolin is a spiny anteater, and these viruses recombined and made COVID-19, and they think it was in pangolin, a spiny anteater, which was taken to China, and from that pangolin they think it infected the first human in Wuhan in China sometime in 2019. Any question about that? Transmission of COVID-19 is mainly from droplets from coughing or sneezing or talking or singing from an infected person, and actually even from somebody just breathing. Very small droplets can come out of the person containing COVID-19. So if you're around somebody who's just breathing and they're infected with COVID-19, you can get it. Most of it is from coughs or from sneezing or from talking or singing. And in fact, singing is one of the worst events that spread COVID-19. So you want to stay at least 6 feet away. They think the droplets mainly travel 6 feet. And that it's mainly breathe in, but it can survive on surfaces, so you can pick it up, and it can survive for a day on paper or cardboard, and for 3 days on plastic or metal. And so when you touch surfaces, it's a good idea to wash your hand to make sure you're not getting COVID-19. Like when you touch your mail, open your mail, and then wash your hands, because the mail can have COVID-19. What I do is when I get mail, I let it sit for a few days, and then I open it, because the COVID-19 will be dead by the time I open my mail. But the point is, when you're touching mail or your groceries, you should wash your hands afterwards, because if somebody who had COVID-19 and they coughed on that, or they touched it, they could put COVID-19 on it. And you know that plastic surface, I don't have a piece of mail here, but the plastic surface on the mail, the COVID-19 can survive on that for 3 days. For a day on regular mail. The symptoms of COVID-19 are very much similar to the flu. Let me see if I can remember. They added 2 symptoms recently, which I don't have here, but I think it was you can get diarrhea or constipation from COVID-19, and I don't remember what the second one was, but mostly they're flu-like symptoms, although runny nose and sneezing are less common with COVID-19 than when the flu. They do happen with COVID-19, but they're just less common. And the mortality is much more common. Another one that's more common is COVID-19. People frequently have shortness of breath and that's because the COVID-19 is hitting the lower respiratory system, and the flu doesn't typically cause shortness of breath. So that's more common in COVID-19. The incubation period, you can have signs or symptoms 2 days after you're first infected and all the way up to 14 days after you're infected. Some patients, of course, never have signs and symptoms, so some patients are asymptomatic, and it's actually a large number of patients who become infected with COVID-19 are asymptomatic. The CDC thinks it's around a third of patients, and maybe as high as 50% of patients, okay? Meaning they're not entirely sure, but somewhere between 20 and 50% of patients will be asymptomatic. The estimated death rate, they're not real sure, but somewhere between 0.7% and 3%. And actually, that's their best guess. If you look at the data, the last I looked at, which I think that's 2 weeks old. So sorry, I haven't looked at that recently. But the mortality rate was actually climbing and was close to 5%. Meaning of all the known cases of people who have become infected with COVID-19, 5% of them have now died. And they don't know why the mortality rate has been increasing, because they expected that as the time went along and they got more patients who were positive for COVID-19, they thought the mortality rate would go down. In reality, it's actually going up, and they don't understand why it's going up. But like I said, they think the mortality rate's somewhere between 0.7% and 3%. But in reality, like I said, the real measurements, it's higher than that now. And they're not sure why it's climbing, because they thought as time went along they'd get more and more of the asymptomatic patients, and that brings down the estimated death rate. Prevention, have hand washing. You should wash your hands for 20 seconds. Wash your entire hands. Keep your distance from people. Keep at least 6 feet away. Don't touch one's own face because you can get infected. And this isn't on here, but I should put it there. And that is, if you're going out in public, you should be wearing masks. And especially true in Arizona, because Arizona has a really high COVID-19 infection rate, and it's getting worse and worse. So I don't know today, because I haven't looked, but like 2 days ago, it was the highest infection rate it had ever been in the state of Arizona. So if you go out in public, please wear a mask. And another thing you should do, like you're going to the grocery store, wear gloves whenever you're touching stuff. And then when you're all done, take off those gloves and throw them away, or if you want to reuse them, alcohol them or put hand sanitizer on the gloves, and then let them dry, and then you can use them. Okay? Any questions about that? If you're sick, stay home, because it could be COVID-19, and wait until you don't have any signs or symptoms before you leave the home. All right, any question about COVID-19? Viruses can cause cancer. Cancer is where human cells are changed, we'll say mutated, and then they're losing their growth regulation, so the cells are continuously growing. Some cancers are actually caused by viruses, and that's estimated to be 10 to 15% of all human cancers. Some cancers are predominantly, if not totally, started by a virus, like human papilloma cancers, cervical cancer, some papilloma skin cancers. That's mainly caused by a virus. Other virus, like liver cancer, frequently caused by a virus. But there are some cancers, like breast cancer and glioblastoma, that are not associated with viruses. Any questions about that? We already talked about that. Viruses can cause cancer in two ways. If the virus integrates into the human DNA, for the cell's DNA, that permanently changes the DNA in the region where the virus integrates, and that can mutate the DNA in that region, and that can go on to lead to cancer. It's a little bit more complicated than that, like when the virus enters the DNA can change the expression of the genes in that region, but we're not going to talk about that. So just if the virus integrates in the DNA, you can mutate the DNA and that can lead to cancer. And that's just a simple way for how viruses can cause cancer. There are also oncogenic viruses where the virus encodes an oncogene. This is a mutated cellular gene involved in cellular growth, and so this oncogene causes the cell to continuously grow, and the oncogene can be brought into the cell and then help cause that cell to become cancerous. Any question about any of that? Meaning that gene causes the cell to continuously grow, and that can lead the cell into becoming a cancer cell. We haven't talked about the latent life cycle. Animal viruses can be following the lytic life cycle or the budding life cycle, but they can also enter the latent life cycle. And generally these are viruses in the budding life cycle, but there could be some exceptions. The latent life cycle does have another life cycle of the virus, like the budding life cycle, because when the virus is in the latent life cycle, the virus is not replicating. The virus is actually inert in the latent life cycle. So it has to have a life cycle where the virus replicates, and that's generally the budding life cycle, and in the budding life cycle is when that virus replicates. Is that clear? So herpes viruses are an example of a virus that follows the budding life cycle, but they can go latent, and the best example for that is chickenpox. When the child has chickenpox, the virus is replicating and causing the chickenpox symptoms, and the virus is in the budding life cycle. But the virus, after the child no longer has symptoms of chickenpox, enters the latent life cycle, and that can go on for many decades. Okay? Where the virus is just hiding out in the cell, and it's the ganglia of the nerve cells where it's hiding out. And it's in the latent life cycle. The virus is inactive, not replicating, not really doing anything except hiding out. And that's what it does in the latent life cycle. But it can come out of the latent life cycle and then cause shingles. And what happens with that is the virus enters the budding life cycle, and the patient then gets shingles, and the virus is replicating, and it's infectious, and it's not in the latent life cycle, it's in the budding life cycle. There's usually a stress that determines when the virus leaves the latent life cycle and enters the budding life cycle, and that can be when the patient becomes immune deficient with old age, or when the patient becomes infected with the cold sores. It can be simply the patient comes down with a cold, and that causes the virus to leave the latent life cycle, and then you can get a cold sore. Any question about any of that?
Yeah, quick one. The latent life cycle is also the lysogenic cycle, right?
No, lysogenic is only for a bacteriophage, for a bacteriovirus. So a virus that's in the latent life cycle is only an animal virus. Most of them replicate in the budding life cycle. So when the virus is replicating, it's not in the latent life cycle, it moves into the budding life cycle. Okay? Did I confuse you?
Sort of, but I'll figure it out.
Okay. There are also persistent viral infections, and these viruses follow a different life cycle, and it's a persistent viral infection life cycle where the virus is mostly leaving the cell. They're naked viruses, and they're leaving the cell in vesicles. So the cell is packaging up the viruses and making a vesicle. It's a membrane surrounding the virus and whatever else is inside the membrane. And then that vesicle fuses to the cell membrane, and that's how the viruses are shed from these patients. And that's the persistent life cycle. And initially, the patient will have the virus at low numbers, but they're persistent. So the virus is always being made by the patient, and the virus load increases slowly over time, and the infection can last for the lifetime of the patient maybe, but for a long period of time. And generally, the virus load gets worse and worse. And so oftentimes they can be lethal. Okay, so it may be fatal. HIV, which actually is a budding virus, can follow the persistent life cycle. The exception is that the viruses are budding. They're not being secreted by a vesicle. The naked viruses would be secreted by a vesicle and then leave the cell by that means. Subacute sclerosing panencephalitis is a case of a persistent viral infection, which I'm sure you've never heard about. There are some viruses that can be tolerated, but they call them persistent. I hate to say that with the chickenpox virus, so I should say we're going to skip talking about that. The only time it would be persistent is when the patient gets shingles, and the shingles never go away. And it's not really the persistent virus infection, because the shingles are only infective for about 2 weeks. And I'm not sure why the patient always have shingles. About 1 in 1,000 patients who come down with shingles, maybe even less than that. The shingles never go away. And let's not even talk about it. But sometimes they're tolerated, while cytomegalovirus, the patient is always secreting the virus, and persistently, sometimes at high levels, and the patient seems to tolerate it. Okay? They're just continuously shedding virus, and if it's being shed at high numbers, you want to stay away from these patients. If you're not infected, about 50% of Americans are infected by cytomegalovirus. And sometimes it'll be a persistent infection where it's continuously shed. Okay? And that's just one that's tolerated. Like I said, oftentimes with these persistent viral infection, the patient will die of the viral infection. And that's why it starts out low, where you're increasing more and more viruses, and then it blooms, and then the patient dies. Okay? That's different from an acute viral infection like the cold. Very shortly after you get infected, you secrete large numbers of the virus, and then the virus load decreases very rapidly, and then you're cured and the virus goes away. Okay, so that's an acute virus infection. That's different from a latent virus infection where when the child gets chickenpox, they have an acute infection, and then the disease goes away. And then many years later, the virus comes out of latency, and then it actually enters the budding life cycle. And then the patient will have shingles, and for the first 2 weeks of shingles, they are infective. And then generally, for most patients, the shingles go away. That usually happens after about 3 months or more. So it's a long time, but they're only infective for about 2 weeks. And the virus leaves the budding life cycle and then goes back into latency. Okay? So those are the 3 types of viral infection. An acute infection, an infection which goes into latency and then comes back out, and most of the time drops away again. And then the persistent viral infection, which tends to increase over time, and in the worst case will bloom, and then the patient dies. And that's frequently what happens in HIV. The patient comes down with AIDS and then dies. Nowadays, they do have drugs to treat that, and so the patient can actually live for a long time with HIV now. All right, any question about any of that? If not, let's move on to prions. Prions are infectious proteins, and that's what the name comes from, proteinaceous infectious particle, and that's what a prion is. And how you get a prion is mainly by ingestion of the infectious protein or by inheriting a defective gene, which when you're young, the defective gene appears to function normally, but over time can fold into the prion protein, and then you can get prion disease. It can be done by transplants or surgical instruments where you're passing on the prions and the patient becomes infected. It generally causes spongiform encephalopathy, where disease is in the brain and you have vacuoles that look like holes in the brain. That's why it's called spongiform encephalopathy. And the best known case of this was in Britain, where they had an epidemic of mad cow disease, also known as bovine spongiform encephalopathy. And around 150 British humans came down with Creutzfeldt-Jakob disease from eating tainted beef. And then around 150 British cats also came down with mad kitty disease, I guess you'd call it, from eating tainted beef, and they also became infected. Surprisingly, British dogs, which were eating the same food as British cats, did not become infected. So dogs were resistant to it, but cats and humans were not, and we had a few cases in Britain where people came down with encephalopathies from eating tainted beef. If you get it in your family, it tends to run in your family, and you know about that because family members come down with prion disease. So that's inherited, and usually the people, when they're young, it's not expressed, and so they go on to reproduce. And then later in life, when the patient is middle-aged or older, the symptoms start appearing and the prion disease forms. And what happens is one of the proteins changes its folding pattern and it becomes the prion protein, and then comes up to the normal protein and transforms it into the prion protein, and then more and more of the prion protein is made, and the patient enters Creutzfeldt-Jakob disease. Okay? Any question about any of that? Kuru was another example of that, where the people of the Fore tribe of Papua New Guinea, in their funeral ritual, they consume the bodies of the dead, meaning dead family members, and when they're eating the brains, it led to them getting prions, and so they were getting prion disease, which was called Kuru. How this happens is that if you're inherited, or even if you eat it, the prion protein comes up to the normal protein and then causes this protein to fold into the prion form of the protein, and then the body says, "Oop, I need more of the normal protein," and so they make more, and then there's more of the prion protein to convert that into the prion protein, and so it just gets worse and worse, and the nerve cells and these stick together and then go on and form the plaques in the brain, and the patient gets prion disease. Any question about any of that? That's how the prion protein replicates. It is the same amino acid as the normal protein in the cell. It's just a different folding pattern. And of course, it's lethal. The patient will eventually die. All right, we talked about plant viruses. They work pretty much the same. They cause diseases of plants, and that's all you need to know about. And then we talked about viroids. Viroids are infectious RNA molecules. Fortunately, there are no viroids known in animals that cause disease, so there are no human viroids that cause human diseases, but there are viroids which cause plant diseases, and the only one I talked about was stunting of potatoes. If the potatoes get infected with a viroid, an infectious RNA molecule, it can cause stunting of the potatoes, and so instead of getting a nice big tuber, you get this stunted tuber. Any question about any of that? If not, I think we'll end here for the review, and come back for the second part. I'll actually be starting the next lesson. I think it's chapter 7 at 2:30. Any questions? All right. I'll see you at 2:30.